It's amazing to think about all the advances happening in bioengineering, like actually growing miniature organs in a dish.
Oh, absolutely.
Pituitary organoids, for instance.
Yeah.
These tiny little 3D cultures that kind of act like mini versions of that really important gland in our brains. Sounds almost like science fiction, right?
Yeah,
But getting them to grow consistently and, more importantly, correctly, that can feel a little bit more like trial and error.
it can. That variability is really a key challenge in the field. You're basically trying to recreate this incredibly complex developmental process, but now you're doing it in a dish in a lab, right?
Right.
And being able to really control that and ensure that every single organoid, every batch, turns out exactly as you expect it to, that's a big hurdle.
It is a huge hurdle. And so that's what makes this deep dive so fascinating because we are looking at some truly cutting edge research that's using some serious computational power to bring more predictability to this process.
Yeah.
Specifically, we're talking about deep learning. Think of it like a smart assistant, but for growing better pituitary organoids.
Yeah.
And we've got a fascinating research paper here that details exactly this kind of AI-powered approach to predict how well these organoids are actually developing.
Right.
And we're going to be extracting the most interesting and important insights for you, giving you a quick route to understanding, this exciting progress.
Yeah, so before we jump into all the technical details, I think it's good to remind ourselves why anyone should care about these tiny lab-grown pituitaries in the first place.
Yeah, good point.
I mean, despite its small size, right, the pituitary gland really is a master regulator. It controls this vast array of bodily functions. It releases hormones, essential hormones that govern things like our metabolism,
Wow.
reproduction, our daily rhythms, even how we respond to stress. That's all tied in through what's called the hypothalamic pituitary adrenal
The
axis.
HPA axis, yeah.
Exactly.
And when this tiny little gland malfunctions, I mean, it can have some pretty serious consequences, right?
Oh, absolutely.
We're talking ACTH deficiency, which can lead to chronic fatigue, loss of appetite, and even life-threatening situations in really severe cases.
Yeah.
I mean, current treatments, like steroid replacement therapy, they can manage these conditions, but they don't always address the underlying root of the problem, right?
That's right. And that's really where this idea of pituitary regenerative medicine comes in,
Yeah.
and where these hypothalamic pituitary organoids hold so much promise, right? Because if we can grow these tissues in the lab and use them to replace or repair a damaged pituitary, that could be a real game changer.
Huge.
And there's actually been some really encouraging work, preclinical studies, showing that transplanting these organoids into mice with pituitary deficiencies can actually improve their pituitary function and even reduce mortality.
Wow.
So that's a huge step toward a future where we might have completely new ways to treat these conditions in humans.
That is exciting. But, and this is what this research we're diving into really grapples with. When you're growing these organoids, you don't always get, like, a perfectly uniform batch,
Yeah,
right?
that's right.
Some of the cells will develop into the desired pituitary tissue, but others might head down a different path entirely.
Right.
And right now, assessing which organoids are, like, high quality, it often relies on the judgment of a researcher, someone with a lot of experience.
Yeah.
But as you can imagine, that introduces some inconsistencies because it's a subjective assessment.
Right. It can be really subjective. So that's really the central issue that this research tackles, this inconsistency, this heterogeneity in these cultures, and the need for a more objective and dependable method to really evaluate the quality of these organoids, especially while they're still developing.
Yeah.
And that's where deep learning comes in.
So let's unpack how they actually did that. What exactly did these researchers do?
So the idea was to basically train a really sophisticated computer model to analyze images of these developing organoids and then predict whether they're actually on the right track to becoming functional pituitary tissue. Interesting. So imagine teaching a computer to visually identify the hallmarks of a successful organoid, like a little quality control expert.
Yeah.
And they use two really powerful AI systems for this. One called EfficientNetV2-S and another called Vision Transformer. Very fancy names.
Very fancy.
Yeah.
Can you maybe give us a little bit of a more accessible way to understand how they built this predictive tool?
Sure. So they started with a human embryonal stem cell line.
Hmm?
Now, this cell line had a really clever built-in reporter system.
Okay.
So certain cells in this line, they're engineered to have this fluorescent tag. Imagine a tiny little light bulb that would switch on only when a specific gene called RAX was active.
Okay, and why is RAX so important?
Well, RAX is a really key player in the early development of the front part of the brain,
Okay.
which is where both the hypothalamus and the pituitary gland eventually develop. And because these two structures are so closely linked during development, seeing RAX activity early on, that could be a good sign that both the hypothalamus and the pituitary are on the right track.
So RX is almost like an early indicator that a potentially successful hypothalamus and pituitary are developing.
Exactly, exactly. They're kind of going hand in hand. And the researchers actually observed that organoids showing strong RAX activity, meaning they were glowing brightly at day 30 of their growth, tended to become much better at secreting ACTH later on,
Right.
which is that crucial hormone we mentioned before,
Yeah.
right? And they also had more ACTH producing cells by day 100.
Okay.
Conversely, low RAX activity at day 30, that was usually a sign that the pituitary wasn't going to develop so well later.
Gotcha.
So that really established RAX activity as a good marker of future success.
Okay, so they had this fluorescent marker to tell them about the RAX activity. How do they use that to teach the AI?
Right. So at day 30, they took all their organoids and sorted them into 3 categories based on how much of their area was showing this RAX glow. Category A, that was for organoids with over 70% glowing bright, you know? Category B, 40 to 70% kind of medium glow.
Okay.
And then category C, less than 40%, the dim ones.
Okay.
And importantly, category C, that included all the organoids that showed low overall RAX activity, regardless of the exact percentage.
So high glow, potentially good, low glow, potentially not so good.
Exactly. And then they took regular microscope pictures of these organoids, bright field images, about 500 for each category.
Okay.
And these images, they became the training data for the deep learning model. So remember those 2 AI systems I mentioned?
Yeah.
EfficientNetV2-S. Think of it as a really efficient scanner for the computer. It looks for very specific visual patterns in the images, things like edges, textures that give it clues about how the organoid is developing.
Okay.
The Vision Transformer, on the other hand, that takes a more holistic view, like looking at the entire structure of the organoid and how all the different parts relate to each other.
So one is detailed, one is big picture.
Exactly. Kind of like how you understand a sentence by looking at all the words and how they relate to each other, right?
Yeah.
And they fed all these images into the AI, along with the information about which category each organoid belonged to based on its RAX glow.
So they're basically telling the AI, this is what a good one looks like, this is what a medium one looks like, this is what a not so good one looks like.
Exactly. And they used really sophisticated training methods to help the AI actually learn the visual differences between these categories and start making accurate predictions.
And once the AI was trained, how well did it actually perform at predicting those categories?
Well, the results were really encouraging, I have to say.
Good.
The combined predictions from both of those AI systems, what we call an ensemble model, it's like getting a second opinion.
Yeah.
It reached an overall accuracy of around 70% in classifying the organoids.
70%? That's pretty good.
Yeah, but what's really interesting is how well it performed with that category C, the ones with the low RAX activity, remember?
Mhm.
Here, the model showed high sensitivity, meaning it was really good at correctly identifying those lower quality organoids,
Okay.
but also high specificity, meaning it was also good at correctly saying that the high quality ones were not in that low quality category.
Gotcha.
And that's really important because if you can reliably identify the less promising organoids early on, you can save a lot of time and resources.
Yeah. You're not wasting time on the duds.
Rose?
So it was particularly good at spotting the ones that weren't developing well.
Precisely. And when they used a more detailed analysis, what's called ROC curves, that further confirmed the model's ability to distinguish between the categories, especially those high and low RAX groups.
So that sounds like a really promising step towards more consistent organoid cultures. But I'm curious, how does this AI actually compare to the human experts, the ones who are, doing this work day in and day out?
Right. So to really test this, they did a really interesting experiment. They asked 3 experienced cell culture experts to try and predict the REX category, just by looking at those bright field images without knowing anything about the fluorescent marker, of course.
Just based on the visual cues in a regular microscope image.
Exactly, just like how they would normally assess their cultures.
Okay.
And the accuracy of these experts ranged from about 47% to 60%, which is pretty good, considering the complexity.
Yeah.
But when they compared this to the deep learning model, the AI was consistently more accurate than all 3 experts in both sensitivity and specificity, especially for identifying those tricky Category C organoids, the low RAX ones.
Wow. So the AI is picking up on some subtle visual cues that even the experts are missing. That's pretty incredible.
Yeah, it really highlights the power of these AI techniques to discern these subtle characteristics that can be really hard for humans to consistently and objectively assess, even with a lot of experience.
And for you out there listening, this could actually mean that future regenerative therapies become more reliable and probably more cost-effective because we'll have a better way to grow the right kind of cells right from the start.
Exactly.
Now, we know the AI can make these predictions, but I'm curious, how does it actually see?
Yeah.
Like, what features in those images is it focusing on to make these
Oh,
decisions?
that's a great question. And understanding how the AI is thinking can actually give us a lot of insight into what really makes a good organoid in the first place.
Yeah, for sure.
So they used some really cool visualization techniques to get a glimpse into how the AI is making those classifications.
Okay.
For EfficientNetV2-S, they used a method called GradCam. And basically what that does is it creates a heat map highlighting the areas in the image that were the most important for its decision.
So, like, showing you what the AI is zeroing in on.
Exactly. And they found that this model often focused on the outer edges of well-formed organoids.
Okay.
And interestingly, also on the fluid-filled cystic regions in the poorly formed ones.
Huh.
And that actually aligns with what human experts often look for as well.
So it's kind of picking up on some of the same visual indicators that an expert would look for.
In some ways, yes, but for the vision transformer model, they used a different approach, and it actually suggested that it was looking at the entire organoid, more broadly,
Right.
paying attention to the overall structure, the denser tissue parts, the edges, any cysts, and even the central region.
So one model focuses on the details, and the other one is looking at the big picture.
That's a good way to put it. It really shows how these different AI systems can approach the same problem from slightly different angles, which is probably why combining their predictions in that ensemble model was so effective.
Yeah.
They're capturing different aspects of what makes a good organoid.
Yeah. Now, one potential challenge for using this in actual medical treatments, and I noticed this in the paper, was the use of that fluorescent RAX marker.
Right,
I mean, you can't exactly go around transplanting genetically modified cells into a patient.
right. Yeah, you're absolutely right. While the Rx.Venus marker was invaluable for training the model and for really understanding the link between that early gene activity and the later development, for this to be used in therapies like transplantation, we'd need a way to assess organoid quality without introducing any kind of genetic modification.
Right. So how did the researchers address that hurdle?
Well, that was a crucial next step in their research.
Yeah.
They tested their model on a different human embryonic stem cell line called KES1,
Yeah.
and this cell line did not have that fluorescent RAX marker.
Gotcha.
So they took the AI model that had been trained entirely on those RAX-labeled cells and used it to classify just regular microscope images of these unmodified KES1 organoids at day 30.
So basically a brand new cell line that the AI had never seen before. And could it still make accurate predictions?
Remarkably, yes.
Wow.
The model achieved an accuracy of around 72% on these completely unseen KES1 organoid images.
72%. That's amazing.
And even more importantly, when they then examined the actual RAX protein levels in the KES1 organoids that the model had classified as, potentially high RAX or low RAX, they used a technique called immunostaining to actually measure the protein.
Okay.
And what they found was that the model's predictions were right.
Wow.
The ones that it predicted as high RAX, they actually had more RAX protein and vice versa.
So it really was picking up on something inherent to the way the cells were developing, not just the presence or absence of that fluorescent tag.
Exactly.
Yeah.
Exactly. It really shows that the model had learned to recognize some kind of general visual features that correlated with RAX activity, even without that artificial marker.
That's a huge step toward those potential clinical applications we talked about.
It really is.
And the story gets even better.
Does it?
Oh, yeah, because when they continue to grow these AKES1 organoids and measured their ability to secrete ACTH at day 100, remember that's the key hormone,
Yeah.
they found a clear link to the model's initial predictions way back at day 30. So the organoids that the AI had classified as high RAX producers, they secreted significantly more ACTH than the ones it classified as low RAX producers.
That's amazing.
And even more, those predicted as high RAX, they stayed well-formed and didn't develop cysts even at that later stage.
So it's not just about RAX activity, it's about the overall quality and health of the organoid.
Exactly. So we've got AI trained on one cell type with a marker that can accurately predict the developmental potential of a completely different unmodified cell line just by looking at some regular images early on. I mean, that
Yeah.
opens up a lot of possibilities.
It really does. It opens a clear path for developing these kinds of predictive tools that don't rely on any genetic modifications, which, as we said, is crucial if we want to use them in therapies like transplantation.
Yeah, for sure. Now, did they actually look at the molecular reasons behind why the model's classifications were so accurate in those KES1 cells? Like, what was it picking up on at that level?
Yeah, they did. They wanted to understand what was going on beneath the surface.
Yeah.
So they took those KES1 organoids that the model had predicted as high RAX and low RAX, and they stained them for a whole bunch of different proteins.
Okay.
And what they found was that the high RAX predicted organoids had significantly higher levels of, well, the RAX protein itself, unsurprisingly, but also other key proteins that are known to be involved in the development of the central nervous system, things like CDH2 and SOX1.
Okay.
And what was interesting was that other markers that are usually associated with the hypothalamus and the oral ectoderm, those didn't show any significant differences between the two groups.
So it really seems like that model is honing in on RAX expression and its downstream effects on the overall structure and the presence of those other really important neural development markers.
Exactly. That's the key insight here, is that RAX expression seems to be a crucial driver of the morphological features that the AI model is learning to recognize.
So let's wrap up by summarizing the major benefits here. What are the key advantages of using this deep learning approach for growing pituitary organoids?
So first and foremost, it provides a really accurate way to predict RAX activity and therefore the overall quality of these organoids really early on in their development.
Yeah.
Second, and this is crucial, it can do this assessment without needing any genetic modifications.
Right.
Remember, that's super important for potential clinical applications down the line.
For sure.
Third, and this is the really impressive part, it actually performs better than those experienced human researchers, particularly when it comes to identifying those lower quality organoids.
Yeah.
And that has huge implications for research because now scientists can focus on the most promising cultures early on, saving a lot of time and resources.
Definitely a game changer in terms of efficiency.
Absolutely. And it also means that you can make decisions earlier in the lab. You don't have to wait as long to figure out if your cultures are on the right track.
Right.
And the paper also hints at some other really exciting possibilities, like combining this model with other AI models that could predict the activity of different genes.
Oh.
So then you'd have this even more comprehensive understanding of organoid quality.
Yeah, like a multi-pronged approach.
Exactly. And it was really fascinating to see that while the AI learned some of the same visual cues that the human experts use, things like the importance of the periphery and the presence of cysts, the vision transformer model also seemed to have its own unique way of looking at things, you know?
Yeah.
It was taking in the bigger picture, which is really interesting.
And it even touched on this really mind-blowing idea of like using images to potentially infer gene activity in living systems,
Yeah.
like that reciprocal prediction idea.
It's almost like science fiction.
Right.
But it really points towards the future of developmental biology and regenerative medicine, you know, where these fields could be heading.
Yeah, yeah. So big picture, this deep dive really just underscores the power of deep learning.
Oh, yeah.
It's a brand new tool
Yeah.
and is really valuable for tackling these incredibly complex processes like growing pituitary organoids. And for me, it was a real aha moment to see that an AI system can actually go beyond just automating a task. It's actually performing better than human experts in this really nuanced area of biology.
Yeah.
And that's pretty remarkable.
It is, and it really makes you think about the bigger implications, like where else in biological research or medical practice could AI step in and potentially improve these processes that currently rely so heavily on human assessment?
Yeah, good question.
Like, think about other organ systems that people are trying to grow as organoids.
Right.
Or even just analyzing those traditional microscopy images in developmental biology where you're looking at how embryos develop.
Right, right.
You know, the paper even mentions this possibility of predicting something like FOXG1 expression, which is a gene involved in brain development, just by looking at images of the developing forebrain.
Yeah.
That opens up a whole new realm of possibilities.
It does. And it makes you wonder, you know, given that AI can pick up on these subtle visual features that we now know are so strongly linked to the function of these organoids, what other hidden patterns are out there?
Yeah.
What other things are we missing that deep learning could potentially uncover in this incredibly intricate world of cellular development?
Right.
It really makes you think.
It does.
What surprising new insights into human biology might we find if we use these advanced analytical tools?
Yeah,
And that's something I'm sure we'll be keeping an eye on in future deep dives.
absolutely. This is just the beginning, I think.
Yeah, the potential here is just huge.
It's really exciting.
So for those of you listening, if you're interested in learning more, I definitely encourage you to check out the full research paper. We'll have a link to that in the show notes. And, this is just one example of the amazing work that's happening at the intersection of AI and biology.
It really is a fascinating field, and it's moving so quickly.
It is. And, who knows what incredible discoveries are just around the corner.
Yeah, I can't wait to see what comes next.
Me too. Thanks for joining us for this deep dive. And as always, keep exploring.